We're talking today to Dr. Laura Esserman, who runs the Carol Franc Buck Center for Breast Care at UCSF. Dr. Esserman directs the Ispy trials. Ispy trials are considered the pathway to personalized medicine and have the potential to significantly reduce the cost of the drug development process and speed up the process by which we bring new, promising drugs to market. Welcome, Dr. Esserman.
Thank you so much.
I want to start the conversation by asking you to explain to a layperson, what are Ispy trials? How do they work? What are the goals?
Well, the goal is very simple. It's to try and find the right drug for the right person and to give it at the right time and to do it now in a more timely way.
So how are they different why is this different from business as usual in terms of the way drugs are typically developed?
So I think there are two big paradigm shifts. And the first is that our standard process for drug development is to test drugs first in the metastatic setting. And those drugs that work, so there's three phases of trials. There's phase 1 to look for safety signal. There's phase 2 to see if there's any evidence that there's an impact or efficacy. 3, it's a head-to-head study to the standard of care.
Okay, and that is often a very expensive, cumbersome, and time-consuming process.
Right, and particularly we do it by first starting the metastatic setting. And I would say that so far in breast cancer, we haven't cured anybody in the metastatic setting, or almost never cure anybody. And then you do these phase 3 trials, but the time where for the drugs that are truly successful, when you move them to the adjuvant or the primary or early stage breast cancer setting, that's when you can actually save lives. So, and it's really thought, well, you have to really test them in the metastatic setting and then introduce them to the early stage setting. Mm-hmm. And there, what we normally do is someone has a large breast cancer or bigger breast cancer, they get operated on, then they're given chemotherapy plus or minus this new agent, and then we wait to accrue the patients, and then we wait for a recurrence. And that can take 5 years. So if you're spending about 7 years developing the drug in the metastatic setting, and then you've got to spend at least 2 years accruing, another 5 years waiting, there is no way to get that knowledge turn to less than 12 to 15 years. There's just no way. So you're set up for that huge knowledge turn of 15 years. You've got such an urgent situation. When I sit in front of a patient who's 39, has bad breast cancer, has 3 little kids, they don't have 10 years to wait. So you're thinking, okay, that's the first problem. Your second problem is that most drugs that get to phase 3 fail. So we're not really doing a good job of screening the drugs that we should be moving forward.
They fail. They fail after having gone through.
So they either don't finish the trial, or they don't succeed, or they don't meet their endpoint. Okay. So then you have to say, well, is really the metastatic setting the best place to even test these drugs? Because it's already a place where you can't really overcome the biology, because you've selected for, cancer cells move and change and develop resistance. So I would submit, if you take the analogy of CML, chronic myelogenous leukemia, Gleevec is the poster child for the biologically targeted therapy. But interestingly, if you give someone that drug in blast crisis, which is our equivalent to metastatic disease, it doesn't work very well. So I would submit that the place to test these drugs are the women who have high-risk disease. So we need to start sooner. And the other thing is, if you want that knowledge turn to be 3 to 4 years, not 15 plus years, you've got to think differently. And so when someone comes in with a bad breast cancer, where you know that person's going to get chemotherapy, surgery is not going to save that person's life. People with breast cancer don't die because the tumor is in the breast. They die because the cells get elsewhere and they take up residence in the lung, the liver, the bone, and replace some critical organ function. That's why people die. So systemic disease, systemic spread is what we're trying to avoid. Now, I'm a surgeon, and I can tell you, I'm not going to save someone's life when they come in if I operate on them first. The most important thing we can do is see what systemic therapies work. And if you give it first and you change the order of treatment, so when someone comes in the door, it's better for them altogether, because if they have a great response, they know it. It gives them their life back. I can do less surgery. I can tailor my adjuvant therapy. I can be thoughtful about it. And if they don't have a great response, I am forewarned and I can look for another trial. I can look for something else. You may not want to see that someone isn't having a good response, but you're better off knowing about it, because then you're going to want to do something about it. So the first thing you need to do is say, what is a good endpoint? You have to have an early endpoint. This is the Andy Grove model, right? Think about early
Andy Grove of Intel, yes.
Of Intel.
Yes.
So this is the model of cranking down the knowledge turns and trying to figure out how to learn as quickly as you can. So that early endpoint for us is complete pathologic response. So we now know that if somebody has a great response, the tumor is completely eradicated, they go from this really high risk of dying, 50%, to down to 10% or less. That's critical. Now, it may not be perfect, but what's your alternative? And the alternative is that 20-year knowledge turn. It's not going to be in that patient's or person's lifetime. You can't learn that way. And then the next thing that we did to try and change this is you think about, how are trials written and how are they written? Every company writes their own. Every investigator writes their own, because that's how people get credit. The companies get credit and they control their development. The investigators are the principal investigator of their trials, and that's how they get credit. So every trial has to be different. But in fact, is every trial different? No. It's standard chemotherapy plus X, right, versus standard chemotherapy. So why do we need a new trial for that? So the whole point of Ispy was to instantiate a standard backbone, a platform trial, where here's the trial never goes down. Drugs drop in, so you can test multiple drugs at once. It's the same backbone. So we reorganized the way credit is given out and the way people collaborate. We also have lots of efficiencies where you have a shared control arm, and you can have up to 3 or 4 arms or drugs in the trial at the same time. So we have a master IND, so all the drugs are listed with the FDA. We have a master IND, and every time we change or drop drugs, the trial never goes down. So it's a much more efficient platform. So most trials take a year or two. Once you have an agreement, you have to write your contract, and you have to get organized, and you write your protocol, and then it has to go through, and then it goes through the IRBs and goes through all the IRBs, and then you start the trial up. And that can take a year or two. And for us now, that's down to 4 months.
So they're much more efficient. That could be one benefit. But
Right.
What about the cost, too? Because that is another thing that we hear so much about, is the expense of the drug development process.
Well, so if you think about, time to market is probably, in terms of the expense, if you can shave years off the time, that's probably the best way to save money and time. And if you so it's better to do a larger phase 1, phase 2 trial. So what we say is, the minute you have safety data, if there's a promising pathway that we think targets breast cancer, we'll drop that drug into Ispy 2. And if it succeeds, then it can move on to phase 3 trial for confirmation. But we're trying to screen. So what we do is we also have an adaptive design. So that's another key feature. So we look at biomarker and imaging guidance. So we're trying to figure out for whom these drugs work, because breast cancer is not one disease. Mm-hmm. Now, everybody says that, but then we treat it as if it is. So we need to know in which biomarker subsets are these drugs likely to work.
And this is the personalized medicine element to it.
This is the beginning. It's the first step. The next thing we need to do is keep iterating and learn to adapt within patients. Right now, we have learned to adapt within the trial. So now, a patient with that might be, we have 10 biomarker signatures that we're looking at that they can be assigned to, that the patient is. Nobody gets on the trial without, a full array. So we know lots and lots about that patient. as we start to see a particular drug working for a particular biomarker subset, the next patient who comes in the trial with that biomarker subset is more likely to get that drug. Over time, what we're going to start to do is then start to figure out, okay, who are the people who are not doing well, and can we move them off and then repeat the process so we can keep iterating for them. That's the way we want personalized medicine to be. What is important is that our threshold for graduation is an 85% chance of success in a confirmatory phase 3 trial. So we're trying to graduate those drugs, and that's our new term, to graduate, meaning that this drug should be moved on to the phase 3. We're trying to maximize the chance that we've picked drugs that are likely to win. So we don't need a huge trial. You need a 300-person neoadjuvant therapy trial.
Because that has been one of the questions about this, is that this is not a big sample size, but you still can remain confident in what you're finding.
The next step, which is the phase 3 trial, right now we have our early endpoint, which is whether or not the tumor goes away completely. We call that pathologic complete response. If that actually was the endpoint for regulatory approval, now it's for accelerated approval, but then you still have to prove the event-free survival endpoint. I think as we start to go on with these trials and show that if you get a pathologic complete response, you do great, that we won't have to have that event-free survival endpoint necessarily, that would tremendously speed up the process of trial development. It would allow you to get these combinations on much more quickly and be able to iterate in a completely different way. So think about it this way. In cardiology, the first trials of looking at statins and so on were, well, okay, if you can, the surrogate endpoints are blood pressure, cholesterol, but what you're really trying to do is stop events like heart attacks and strokes. And so these big trials really demonstrated that these drugs stopped all that. So now the endpoint of most trials is blood pressure and cholesterol. That, I think, is the right paradigm for us.
Okay.
And I think both the EMA and the FDA are looking at that. Increasingly, we've looked, there are more trials, especially in the HER2 space. This is a particularly aggressive cancer that we now have targeted therapies for. And it's now, as more of these trials come to maturity, we're learning that those early endpoints indeed work very well and are very predictive of overall success. So that gives me hope that we're going to be able to continue to shorten that timeline. There are so many interesting and exciting drugs out there in combinations, but no one's going to take the time and effort to test them, and people are going to be afraid, right? They're going to be afraid of failing because they don't want their drug to fall off the map if it doesn't work that way. And, it's a very risk-averse culture.
Right.
And it's this total negative spiral that just conspires to increase cost and increase the time.
Well, one of the most remarkable things about these trials is the collaborative nature, and you have so many different institutions involved, the FDA, the NIH, biomarker consortium, drug companies, just to name a few. How do you balance?
And at any given time, we've had up to 20 sites collaborating.
Right, exactly. So geographic differences too. How do you get all of these stakeholders involved, and how do you incentivize them to participate and also make sure all their needs are met? Because this is a very risk-averse culture, worried about credit, and how do you manage that?
Well, we were lucky in Ispy. We picked the leaders who were very forward-thinking and innovative and asked them to join. And people were so excited, really, to have the opportunity to do new things and to try something different. And the culture actually, you have to create a culture of innovation in order to innovate. And that's a lot of what the work has been in Ispy too. We have 9 working groups. There's chairs of the working groups. People have opportunity. There's the biomarker working group that Laura Manfier runs. There's the Agents Committee that Doug Yee and myself run. He's the head of the Cancer Center at University of Minnesota. Angie D. Michelle is head of our operations working group, and she's at University of Pennsylvania. We have our chaperones group. We actually encourage everyone to go out and look for interesting drugs. And if you can find them, instead of having to be the PI of your own trial, the chaperones, are like the PIs of each little arm that comes through. Everyone needs to have skin in the game. Everyone has to get something back out of it. So I'm not the first author in any of these papers. I don't present the results because that's not my goal. My goal is to create a collaboration where everyone gets something for it. And the idea is we tried to make it simple so that if you come in, anyone who's got a tumor that's bigger than 2 or 2.5 centimeters, we know, why are we ever doing an adjuvant therapy trial anyway? We shouldn't be doing that. Why would you not want to integrate that early endpoint anyway? So the order of therapy doesn't change your outcome. It's not better or worse. It just changes the information that you get. So that information is key. I don't ever think we should be doing an adjuvant therapy trial because you're basically you're blinding yourself to the very information you need to help make a decision. At the worst, it won't help you. And at the best, it'll direct everything. So why would you not do it that way? And if you aspire to having rapid knowledge assurance, you have to have that information so you can make better decisions, more thoughtful about which agents to pick. Now, every company doesn't want to do that. Every company is not interested or willing to put their drugs in, and that's fine.
But the ones that you have, as you said, handpicked
so
leaders
Genentech,
from...
Amgen, Merck, some small companies have put their drugs in.
And they're willing to fund these trials, too.
And now they're willing to fund it. At the beginning, we didn't have a regulatory path forward. And we basically got the drugs and then paid for the trial with philanthropic funding. Janet Woodcock at the FDA, I think, who's incredibly forward-thinking, and Rick Pazder as well. We actually talked to them, and I said, look, that they wanted to see more pre-competitive collaboration. So I said, okay, you help me create a regulatory path forward, I'll create pre-competitive collaboration. And we have this small, we've done this through a series of small think tanks where people come to the table and everyone looks at it and everyone, not too many people, but you say, okay, nobody leaves the room. We know we have a problem. No one leaves the room. Until you have it usually in a beautiful place where people can see how beautiful it is outside, say nobody leaves until we come up with a solution.
Right.
And, and then I had people like David Diltz come down and sort of demonstrate, what do you get out of pre-competitive collaboration if it really works that way? The industry learns. Most people have a lot of the same drug classes in their portfolio. And so when they learn from one or the other, they're not all the same, but they get a pretty good indication of what's really going to be important and what's not. And so it helps them and it helps the industry. There's so much of this learning that's underground that everyone's repeating and putting hundreds of millions of dollars in, and it's not getting out to the public. It's not getting out to the industry. Who's the big loser there? Well, the patients, for one, and two, actually the industries themselves. It's not helpful to them either. And, if in fact we can get, short trials of $5 million, most small biotech companies cannot afford $100 to $200 million for a trial, but they can afford $5.
Well, you've actually mentioned in your research that there are other diseases that could benefit from an Ispy-like trial, and this is cystic fibrosis, pulmonary fibrosis, other autoimmune diseases. What will it take for this to be adapted by the industry? And you've pointed out to a lot of benefits to patients and to the industry itself.
Well, I think we have to figure out how to scale. We're working on the IT infrastructure to figure out how to scale that, number one, and build the, where you can build the analytics into the way you collect data. I'll come into this whole idea of one source and the idea of data plans. That's an important way to streamline trials. So we are actually actively looking at the opportunity to replicate this in the myeloma space. We're going to call it MySpy. That's what the myeloma group wanted to call it. But you could imagine that colorectal cancer is a great place. This is something where if you change the order of therapy, that, neoadjuvant chemotherapy, especially for rectal cancers, this means that you can have rectal sparing surgery. People do much better. You can imagine this would be a great way to drive innovation there. I think what we have to do is make it so that this kind of infrastructure works, that it's where people get credit for this. And I think if we are successful and people can start to see these drugs starting to move forward and seeing how fast we can get these drugs to market, then I think it will change. So one of the things that we've been thinking about doing is trying to not only do more I-Spy more, but to also think about, well, how do you link the phases of trial development? Phase 1, phase 2, phase 3, what really makes things fast. There's a lot of white space or sort of wasted time between phase 1, phase 2, and phase 3. And the FDA has been very interested in us trying to have a standing or platform trial for phase 3, where and a lot of this is about standards, right? So you want to do certain things all the same so that you can innovate on where it matters the most.
Right.
So you have to do the surgery the same, you have to do the pathology in the same way, so people have to show that they can harmonize the way that they're collecting the data. Not so much, you don't need every last data element. You need specific data elements, and we don't need to, the amount of information we collect is mind-numbing and so expensive. That is also not necessary. So we're trying to advance the idea of having a data plan, so that here's what we collect. This informs our primary endpoint. We'll collect that carefully, and the rest we'll collect, but we're not going to clean and do all these things that cost so much time and money.
Even the failures to inform you about.
Absolutely,
yeah.
Absolutely.
Okay.
So around the world, we've collected a consortium of people around the world who are willing to put their patients on these trials, because you can imagine if you go seamlessly from the phase 2 trial to be able to drop that drug right into a running trial that's ready to go, you could probably get those patients accrued in a year and be at market if you imagine that. And one of the things we've had is it's been hard to get the phase 1 trials with the safety data with the Taxol backbone or with the backbone. And so we've actually set up our own network of Ispy investigators. A number of them run phase 1 trials, so that we know that when they're running it, we now have 2 or 3 trials in phase 1. So the minute they've reached a dose that is safe and they're expanding it, we now can be writing the protocol, getting the contract together. So that saves 4 to 6 months. So you're further, and then you save another 4 to 6 months pretty soon. Even on top of the 4 years, you've saved another 2 years.
Right.
That is really what it's supposed to be about. And we're doing it in breast cancer because that happens to be the field where I'm an expert, not because that's the only field it can work in.
But
it's a very generic process. And you see the AGILE trial for glioblastoma has been developed. They're working with Bayesian adaptive design, Don Barry, who does all the statistics and the Bayesian design, with all the simulations and all the modeling up front. So all the planning's up front. And if you hit that endpoint, then you know you move on. There's a development in the Alzheimer's field. A lot of people are starting to think about it. But I think the key here is changing our culture, and changing our culture where we take a step back and say, what is the purpose of all this? The purpose of this is to identify drugs that help women with a particular biomarker subset the most, and get those as quickly as possible to market. It's not about publishing, it's not about getting a grant. So I think all of us in the industry have to think hard about how we change incentives to enable these kinds of models to flourish. And that's up to the academic leaders. So I think that the problem isn't technology, the problem is culture.
Well, and it's interesting, and it's a segue into my next question, because one of the criticisms is that you may be getting rid of drugs prematurely if you're setting the bar too high in a way, and just hearing what you're talking about with the increased agility of this kind of trial, the fail fast mentality that we hear so much about in Silicon Valley and in innovation centers across the United States. How do you respond to that? Are you setting the bar too high?
So we aren't actually telling people to discard their drugs. And it's funny, people are saying, oh, if I don't meet graduation, then people are going to think my drug failed. I said, no, we're setting a bar high because we know that you have to have at least a 20% difference in your change in pathologic complete response rate to see a change in the event-free survival rate. So it has to be that high. You know, you may decide, well, maybe I need that drug in combination with something else, or maybe it's close enough, it's 70%, and I'll still move it forward. We're giving back information that's helpful. But, honestly, if you have 10 drugs in your pipeline, and one of them is 80% and the other are 30%, which one do you want to develop? And, how quickly could you figure this out? How could you really organize it? If more people were willing to take a chance, we would do better. I mean, if you take a step back and say, well, how have we done here? If 70 to 80% of drugs are failing in phase 3, we have to just be honest. I think what happens, the reason why people don't want to take risk is they're often not willing to face reality.
So it goes back to changing the culture, as you talked about.
It's changing the culture.
Yeah.
One of the oncologists said to me at some point, I don't like this neoadjuvant model. I don't like to see it when people aren't responding. It makes me uncomfortable. I said, good. Then maybe you'll be excited about giving them a different drug or a different treatment.
Right.
It's like if you do the same thing over and over again, you can't hope for a different outcome. Mm-hmm. What we are doing today is not good enough. Spending $2 billion, taking 10 to 12 years to get drugs to market, how is that a good thing? It is not. Now, I'm not saying this is the only way to do things. I'm saying this is a better way, and it should have its day in court.
So we've talked a lot about this from the industry, the academic, the research perspective. What about the patient perspective? What is their response to this idea of personalized medicine, and what are you hearing from the people who are in these trials?
Well, we don't have trouble getting patients. Like at least half, what is the average rate of getting patients on trials? Like 5 to 10%. With these trials, of all the patients who go on neoadjuvant therapy, half go on a trial. If we say, look, I'm doing this anyway, why not participate in one of these trials? Because we're going to do it this way anyway. I'm going to give you the systemic therapy first anyway. Mm-hmm. Most people are excited about that.
Right.
Patients are not the problem. They are not the problem because you know what? They are in a hurry. They don't have
to
10
save
years
their
to
life.
Wait.
Right.
Or they're worried about their mothers, their sisters, their daughters, their friends. They aren't the issue here. This trial, by the way, is very patient-centered. Mm-hmm. I'm thinking about how to organize the care and change my process of care so I learn as I go. Mm-hmm. They get that. When you stop and explain it, they're like, oh, that makes sense to me.
What might their treatment be like if it weren't for Ispy?
Well, they would you can get standard therapy and you can get it off trial, mm-hmm. or you can get it on trial. But on trial, you might get that plus something else. Something else that as I say, trials are the way we test agents that we think will be part of the future of breast cancer, but we don't know. And we're trying to learn which work. There's informed consent. People have a choice. Mm-hmm. They can do it or not. I'm a big believer that if you fully inform someone, they can make very good choices. And we have not had trouble getting patients to want to participate in these trials.
Phase 1 of Ispy is done. Phase 2 is ongoing. What is the future for Ispy 3? What are we looking at right now?
So Ispy 3 is designed as an international consortium. We actually have designed the trial and gotten the backbone of the trial approved by the FDA. And there's some really important innovations in that process as well. It has to be open label. That's actually not the way most trials are run. But if you're testing novel agents, they all have, in personalized targeted agents, they all have side effects.
Open label.
Meaning it's not blinded to the investigator.
Got it, okay.
And our point is it's not safe because you have to manage the side effects. Mm-hmm. 2, it's not realistic because, if you have a drug that causes diarrhea and people get a lot of diarrhea, guess what?
You want to know.
Right. If you read how and the band played on, this is you learn from the communities, from the AIDS communities, et cetera. This is not some great surprise here.
Right.
And so that means you have to have a lot of security that your primary endpoint is going to be accurately collected. So that means that what we can do is we have 2 pathologists trained in the method of carefully evaluating the tissue. So you have really careful trials and everyone doing that piece the same. And we are creating this sort of what we call one source app so that people can just fill in the key pieces of data. They won't know what drug they're on or what arm they're on, and they can fill that information on. So this actually brings up an important issue. One of the culture changes in any kind of adaptive trial is that you have to get the data in real time. That means right then. That's not what we're used to. And so the more we need to get used to getting data in real time, the more we can start thinking about, why don't we enter the data we really need as a checklist? Because that actually improves our care. So one of my big issues and big areas of interest is trying to build these, what we call one-source checklists. And you can imagine if you have the mission-critical data you need to care for someone, that's also the data you need for trials, for registries, for quality improvement. What if next year a drug comes out for such and such, you'll be able to pull, okay, I want to see all my patients who had this. Anyone from the business industry is like aghast that we don't have that in medicine. And there really is no reason for it. I myself am aghast that I've been working at it for 25 years and since I left business school and don't have it either. What we need is Moneyball for medicine. I laugh and think about all the information everybody has on their fantasy players.
Right.
And we should have at least that set of data on the patients we treat when so much is at stake. And that, again, could change by what we're paid to collect. But if you imagine you have these kinds of data at real time, it would not be so hard to put people on trials. Click, that data is collected, goes to the trial, that goes to trial matching, trial eligibility. And when it's one source, or electronic source, means the physician has approved it and looked at it, it doesn't have to be recollected by these trial monitors. All of the data management in trials is what is so unbelievable. That's half the cost of trials. All that could go away if we actually made the improvements in clinical practice that we need.
In real time.
In real time. So I always say, we're in clinical practice. What are we practicing for? We should be practicing to get better, right? But how can you get better if you don't get feedback?
Well, this is my final question for you, and this is that you're painting such a vivid picture of the beginning of personalized medicine. What does the future hold? Is it this Moneyball for...
I think it is. In my book, so my mantra has always been, for the last 20 plus years, you should be tailoring your treatment to biology, patient preference, and clinical performance. But you have to get the information on all of that. On biology, these are the basic facts we get. If someone comes in with breast cancer, we know there's a spectrum of breast cancers from very indolent to very aggressive. You need to know what that is, and you need to tailor your treatments accordingly. You need to offer people options, and you need to understand what's important to them. Everybody is different. We need to know more about their host genomics. We need to know about their own personal performances, what their background of health is, and we have to know at a moment's notice how our own clinical performance works and not be afraid. And if you want to be an advocate for your patient and not your specialty, you have to know how you perform because that gives you the opportunity to make it better, which is what our patients want, right? So that's how those are the kinds of changes that we have to make. I hope that will happen in my lifetime. It could. I'm going to do everything I can to make it happen. But there's so much at stake here. But you can't, without the tools for quality improvement, we will never unleash the power of personalized medicine. I think one of the big barriers to personalized medicine is that we actually have a system of research and we have a system of care. There's no reason for them to be separate. They have to be integrated because the researchers can't ever get the data. That's the big barrier. And the physicians and the clinicians don't get feedback on what they do. We know that's the sine qua non to making things better, right? So that is how we're going to make change. And we should make it so that these opportunities and these trials, that at least 50-70% of your patients should be on these kinds of trials. Imagine how fast we could learn. Imagine the opportunity. There are hundreds of drugs in the pipeline. How are we going to test them? How are we going to know? How are we going to figure out how to combine them? We have to do better. We have to figure out the ways to speed things up. And that may mean different ways of collaborating, different ways of getting credit, all these things that we're talking about. This is the beginning of all that.
So it's changing the culture.
It's changing the culture. And I'm already looking at our trial design and saying, how can I make it better? As we start to succeed and we get more and more drugs doing better, we said, okay, well, let's pick a new standard. Get that for your intro or your beginning. If 50 or 60% of people have a great response, move them off, and then take these people, set the bar back to zero because they don't have a PCR. Now you can again start testing how to improve their lot. That to me is the essence of personalized medicine. And your trial should anticipate the way you want practice to be. And that's what you want. You try something, you look, you have your solid surrogate endpoint. If that doesn't look good, switch. That's what people should want. And in fact, that's what happens in leukemia. We should really aspire. It was like I say, look, we should aspire to the iPhone 8.
Yes.
Right?
Right.
I'm running a big trial of personalized screening versus not, because I'm trying to understand who's at risk for what kind of cancer. All these things are going to help us merge, who needs to be treated with what, with who's at risk for what, to really get us to a place to even be thinking about prevention, right? And people say, oh, you can't change screening. I'm like, really? Really? If I went in and looked at the phone that I had when I was an intern, which was this giant box that,
Right.
Would anybody buy that? No, it's a relic for a museum. We in our lives, we demand improvement and change in technology. And now when it takes a nanosecond to connect, we're all frustrated. I want to see more of that frustration and demand for change. And thinking about it, we need innovation around healthcare value, around personalized medicine. And if we don't fix our clinical care systems, we're never going to get it. And that is the task at hand. Our clinical care and our clinical trial systems must change and improve. And this is a good start. It's not the end all be all. We need to continue to innovate and improve. But it's definitely a great step in the right direction.
Great. Well, thank you very much.
You're welcome.
That was Dr. Laura Esserman, who directs the Carol Franc Buck Center for Breast Care at UCSF. Thank you.